might seem crazy what I'm about to say. Sunshine, she's here, you can take a break. I'm a hot air balloon that could go to space with the air, like I don't care, baby, by the way. Because I'm happy. Clap along if you feel like a room without a roof. Because I'm happy. Clap along if you feel like happiness is the truth. Because I'm happy. Clap along if you know what happiness is to you. Because I'm happy. Clap along if you feel like that's what you want to do. Here come bad news, talking this and that. Well, give me all you got and don't hold it back.
After the discovery of mitosis in 1875 and meiosis in the 1890s, scientists began to see the links between the behavior of chromosomes and the behavior of Mendel's hereditary factors. They noticed that chromosomes and genes were both present in pairs within diploid cells. Homologous chromosomes and alleles separate during meiosis, and chromosomes and genes both become paired again during fertilization. This led to the realization that genes made up chromosomes, and the formation of the chromosome theory of inheritance. This theory explains that Mendelian genes have specific loci, or positions, along chromosomes, and it is actually the chromosomes themselves that separate and become independently assorted during meiosis.
The behavior of homologous chromosomes during meiosis explains the segregation of alleles to different gametes. The behavior of non-homologous chromosomes explains the independent assortment of alleles for genes located on different chromosomes. This cross between two true breeding pea plants shows how Mendel's discoveries correlate with the behavior of chromosomes during meiosis.
This diagram shows the chromosomal basis of Mendel's laws. It allows us to correlate the results of one of Mendel's dihybrid crosses with the behavior of chromosomes during meiosis. You can see it shows the P generation, which is the parental generation, the F1 generation, the first generation of offspring, and the F2 generation, which is the offspring of the F1 offspring. In the P generation, Mendel started with 2 true breeding pea plants, and will follow them through the F1 and F2 generations. The 2 genes that specify seed color, allele big Y for yellow and allele little y for green, and seed shape, allele big R for round and allele little r for wrinkled. These 2 genes are in different chromosomes. Peas have 7 chromosome pairs, but there's only 2 illustrated here. So you can see on this side is the yellow seeds, and then over on this side is the green wrinkled seeds. So they fertilize, which occurs here, and then the F1 offspring are produced. In the F1 offspring, you can see the law of segregation occurring. The 2 alleles for each gene separate during gamete formation. As an example, follow the fate of the long chromosomes carrying big R and little r. So here at number 1, the big R and little r alleles segregate at anaphase 1, yielding 2 types of daughter cells for this locus. At location 2, right here, each gamete gets 1 long chromosome with either the big R or little r allele, and then here are the gametes. The law of independent assortment is also shown. In the law of independent assortment, alleles of genes on non-homologous chromosomes assort independently during gamete formation. As an example, once again follow both the long and short chromosomes along both paths. So here at location 1, alleles at both loci segregate in anaphase 1, yielding 4 types of daughter cells, depending on the chromosome arrangement at metaphase 1. Compare the arrangement of big R and little r alleles relative to the big Y and little y alleles in anaphase 1. Here at location 2, each gamete gets a long and a short chromosome in one of 4 allele combinations. And then in the F2 generation, the fertilization results in the 9:3 to 3:1 phenotypic ratio. You can see right here the fertilization among the F1 plants yielded 9 yellow round seeds, 3 green round seeds, 3 yellow wrinkled, and 1 green wrinkled. And the fertilization recombined the big R and little r alleles at random.
The first real evidence matching a specific gene to a specific chromosome was discovered in the early 20th century by Thomas Hunt Morgan. So, our next topic is going to be Morgan's experiments. Morgan began by choosing a Drosophila melanogaster, a species of fruit fly, for his experiment. He chose this because fruit flies produce large amounts of offspring very quickly, making them convenient for genetic study. The fruit fly also has just 3 pairs of autosomes and 1 pair of sex chromosomes, which are simple and easy to study under a light microscope.
After years of tedious inspecting of thousands of fruit flies, Morgan discovered a male with unique white eyes instead of the normal red. Morgan assigned names to the eye color phenotypes he had discovered. He called red eyes, the color normally found in natural populations, wild-type eyes. He called the unnatural white eyes a mutant phenotype. He then invented a notation for symbolizing the alleles. The symbol comes from the mutant type, so the alleles for white eyes is symbolized by W. The superscript plus identifies the allele as the wild type that trait, so W plus is the symbol for red eyes.
Now Morgan began his experiment. His parent generation consisted of a white-eyed male fly with a red-eyed female. The F1 offspring all had red eyes, which told Morgan that the wild-type allele, which is red eyes, was dominant. In the F2 generation, Morgan observed the classic 3:1 phenotypic ratio. Surprisingly, though, the white-eyed trait showed up only in males. All of the females had red eyes, while both traits showed up among the males. This led to Morgan's conclusion that a fly's eye color was somehow linked to the sex of the fly.
As you know, a female fly has 2 X chromosomes and a male has an X and a Y. Morgan realized that because the white eye color only showed up in males, the gene was located exclusively on the X chromosome with no corresponding allele on the Y chromosome. Figure 15.4 shows the reasoning behind these ideas.
Thomas Hunt Morgan wanted to analyze the behavior of 2 alleles of a fruit fly eye color gene. In crosses similar to those done by Mendel with pea plants, Morgan and his colleagues mated a wild-type red-eyed female with a mutant white-eyed male. All of the offspring of this cross in the F1 generation had red eyes. He then mated the F1 red-eyed female to an F1 red-eyed male to produce the F2 generation. The F2 generation showed a typical Mendelian ratio of 3 red-eyed flies to 1 white-eyed fly. However, no females displayed the white-eye trait. All white-eyed flies were males. All F1 offspring had red eyes, so the mutant white-eyed trait must be recessive to the wild-type red-eye trait. Since the recessive trait, white eyes, was expressed only in males in the F2 generation, Morgan deduced that this eye color gene is located on the X chromosome and that there is no corresponding locus on the Y chromosome. Because the gene is found exclusively on the X chromosome, a male fly would only need 1 copy of the mutant allele to have white eyes. A male only has 1 X chromosome, so there can be no wild-type allele present to mask the recessive allele. A female, however, could only have white eyes if both of her X chromosomes carried the recessive mutant allele, which was impossible in Morgan's experiment because all of the F1 fathers had red eyes.
Morgan's finding of the correlation between a particular trait and an individual's sex provided a support for the chromosome theory of inheritance. It provided evidence for the idea that a specific gene is carried on a specific chromosome. This concept is crucial to understanding the chromosomal basis of inheritance.
For a review of this section, Concept Check 15.1 can be found on page 289. Question 1. Which one of Mendel's laws relates to the inheritance of alleles for a single character? Which law relates to the inheritance of alleles for two characters in a dihybrid cross?
The answer is the law of segregation relates to the inheritance of alleles for a single character. The law of independent assortment of alleles relates to the inheritance of alleles for two characters.
Question 2. What is the physical basis for each of Mendel's laws?
The answer is the physical basis for the law of segregation is the separation of homologs in anaphase 1. The physical basis for the law of independent assortment is the alternative arrangements of homologous chromosomes in metaphase 1.
Question 3. Propose a possible reason that the first naturally occurring mutant fruit fly Morgan saw involved a gene on a sex chromosome.
One possible answer is, to show the mutant phenotype, a male needs to possess only one mutant allele. If this gene had been on a pair of autosomes, two mutant alleles would have had to be present for an individual to show the recessive mutant phenotype, a much less probable situation.
If you know what happiness is, you